Welcome to your last lecture for Cycle 3. This is the last lecture that will be included in your October term test, Genetic Variation Due to Mutation. I sent a October term test document out this morning, so if you haven't had a chance to check your email, that's okay. It's posted as an announcement. Please read that document very, very carefully. It provides you with detailed information about the October term test. Read it completely and carefully so you can arrive prepared. One important thing I want to mention is that I don't want a single one of you to forget to write your name and student number on both the test paper and the Scantron sheet, and write your group number, names, and signature on the IFAT cards. And you must be saying, yeah, okay, we'll write it. Here's why I make this announcement, and I really, really want to make sure not a single one of you out of 1,500 misses your name. If 1% of the class forgets to write their name on the test, that's 15 students. It's not unreasonable to say that 99% of the students will remember, but 1% will forget. It's not unreasonable, but that's 15 students. And it's a Scantron. You have no name, no student number. Now I've got 15 students. Which Scantron belongs to who? We don't know. Unfortunately, all of them get a 0. That's the moment in life when you look at that moment and you say to yourself, life sucks. Life sucks, because I know I got over 90 on this test and I ended up with a 0. Life sucks. I don't want a single one of you to go through this because it tortures me more than it tortures you. My whole philosophy is to drive information in you and score you a high GPA, and then I have to give you a 0. Tell me why we couldn't work it out and say, okay, you can write a makeup, or you can get consideration because you forgot. Tell me why I can't do that. Put yourself in my shoes. I give you the respect to always put myself in your shoes, and that's how I teach the course. If I was a student, would I want that? No, I wouldn't. Would I want lecture recordings posted so I can go back and listen to what she said and take notes? Yeah, I would. Is it a lot more effort for the prof? Yeah, but that's okay. It benefits you. Would I want to know what I'm being tested on in a lecture, outcomes? Yeah, I want to know that. Do I want to transition from pedagogy to andragogy? Yes. So these are all the things that we embed. Do I have to be a master perfect to be a master? No. No, I don't. So these are all the things that we try to implement. So why do you think I can't do that? Doesn't that create a loophole in the system? Students can walk in there, look at the exam, and go, oh my God, it's brutal. I don't know the answers. I'm going to, quote unquote, forget to put my name so I can get consideration to write and re-weigh my marks. We can't do that. So please, 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 I'm begging you, when you walk into the exam, all you have to think is name, student number, name, student number, name. Just keep repeating it and write it down. Please do not forget. It's a Scantron sheet. We don't know who it belongs to. You can't walk in there and go, oh yeah, you know the one with the 100%? Yeah, that one's mine. You can't do that. You can't do that. We don't know who belongs to who. So please, 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 I'm begging you, do it for me, because it would kill me. It would kill me to give you a 0, all right? We had some questions on meiosis. What do we have to know about meiosis on the October term test? You don't have to know all the nitty-gritty details of meiosis. We're going to cover meiosis in the course. But I'm not going to go over it. This is meiosis 1. This is exactly what happens. Now let's look at the chromosomes. That you're expected to read in the textbook. But for the exam, you have to have an idea of ploidy during meiosis. You have to know the coefficient of C and how it changes. So C and N during meiosis are where the questions are going to come. I'm not going to ask you nitty-gritty details of homologous recombination and what exactly is happening during the anaphase of meiosis 1 versus meiosis 2. No, those are not the types of questions that are going to be on the exam. N and C with respect to meiosis. Okay, so today's lecture is on genetic variation due to mutation. I hope that all of you can appreciate, again, that mutation is not a bad thing. A mutation is often a very good thing. It's involved in evolution. If you look at our genome sequence and look at the way the genes are aligned in all different species, it provides you a lot of information when you're studying evolution, when you're studying genetic variation in a population. These are mutations that have benefited us. So we've evolved, they've been selected for. So mutations are not always bad. Are there bad mutations? Of course there are. They lead to diseases. But there are good ones and bad ones. And sometimes what may look like it's a bad mutation, there's a base pair change, what's going to happen now? It depends where the change is in the genome. It depends. Sickle cell anemia, there's a single base pair change and you get sickle cell anemia. So it just depends where it is. That single base pair change in another region is simply called a SNP, or a single nucleotide polymorphism. And we all have SNPs that are different from each other. So it just depends where that mutation is. You say, it's just a point mutation. It's a big deletion mutation. Therefore, it makes a difference. No, it just depends on the location. If it's in a very important area of a protein that is involved in its catalytic function, then you're going to have some deleterious effects. If it's in the part of an intron that doesn't have splice sites or is not involved in splicing of the pre-mRNA, then it doesn't matter. So it just depends where that mutation is, right? So we get genetic variation due to mutation. So now this lecture will cover the different types of mutations and how we get genetic variation due to this process. Independent assortment during meiosis is one way, and recombination. And I think all of you are familiar with both of these mechanisms, and we'll learn that more in cycle 4, but today's lecture is mutation. October term test document is out there. Please read it and make sure you arrive prepared. So just like you made a list of mechanisms to generate sameness in the genome, you have to make a list to generate genomic diversity. I'm not going to tell you all of them because we're going to learn a lot of it today. So between the first lecture in the cycle 3 and the second, together you should be able to make this list. The list can be longer, but at least you should be able to come up with 3 mechanisms. They can be specific mechanisms on how the cell introduces difference. So how the cell maintains sameness and how it introduces difference. Because remember, we talked about sameness and difference. You need a nice balance. How much human variation is there? In other words, if I'm sitting here and there are people around me, how different is my genome compared to theirs? How different is your genome compared to the 2 people sitting side by side? So this project, that 1,000 Genomes project, in 2015, they had over 2,500 genomes sequenced, 26 different populations. And they tried to sample various different populations, not just one area of the globe. 0.14% in general, 0.14% of your genome is different from their reference genome. They have a reference genome. That's why I say roughly, in general, because they've created a reference genome based on these 2,500 genomes. And so it's 0.14 difference. Chimps, 1.4. You're 10 times different from a chimp than you are to the person next to you. So what is this difference? Is it that you don't have a gene for eye, but this person does? Is it that you don't have a gene to produce bone marrow cells and this person does? Majority of these mutations are SNPs, single nucleotide polymorphisms. And when you spit in a tube and send it to 23andMe, that's what they're looking at, SNPs. Known SNPs, known areas that can provide you information about susceptibility to disease, that can provide you information about other aspects of your health and ancestry. So that's what they're looking at. They're not sequencing your entire genome. They're looking at that difference, the areas that are different, they're known. They're known. In addition to SNPs, there are about 2,500 structural variations. These variations are different than SNPs in that they include mobile elements, copy number variation, and inversions. And we'll touch on all of this, not all in this class, but we'll continue this in cycle 4, but please understand that this is where we're going to be heading in the next couple of lectures. So today we'll cover mobile elements and SNPs and other types of mutations that lead to SNPs. About 12,000 variants of all the variants in your genome are in coding regions that code for proteins. About 100 variants are de novo. De novo meaning that neither one of your parents has it. You have it. You have it though. But you didn't inherit it from your parents. How do you think you yourself got these mutations? Think about it. Well, I didn't inherit it, so how do I have these mutations in my genome? I'm pretty sure all of you know the answer to this. And after today's lecture, I think every single one of you will know. About 30 variants are associated with disease. So a lot of these variants, these mutations, i.e. a double-stranded change, are not associated with disease. Look at all the variation you have. Look at all the variants. But only 30 of them are associated with disease. And I use the word associated. And again, I keep going back to 23andMe. All they can report to you is how much of an association that SNP region has to this disease. This SNP has been associated with heart disease. And if you have this particular mutation in this particular region, you have an X% chance of developing heart disease. And so that's what they're basing it on. It's just an association. It doesn't mean that if you have that SNP, you're getting heart disease. Diseases are multifactorial. If you can say that to someone, you sound like you really know what you're talking about. Oh, I've got that SNP, that's it, I'm done. I got the disease. They're multifactorial. Yes, are there areas of the genome where a single mutation can lead to deleterious effects? Yes, cystic fibrosis is an example. Cystic fibrosis, you don't need to have multiple SNPs everywhere and multiple mutations. All it takes is one mutation in the CFTR gene and boom, you have cystic fibrosis. So please understand that mutations can be good or bad. When they're bad, it depends on the location and the type of effect it's going to have on expression of that gene. That's what it depends on. You can't lump them into two categories, good and bad. Bad means disease, good means you're going to evolve and become more fit. It's much more complicated. If it was that simple, we would have the cure to every disease humanly possible. So let's look at mutations. Let's drive right into the DNA and look at the mutations. What are some types of mutations? These ones you should be familiar from high school: substitution, deletion, insertion, inversion. Deletion, insertion are also called indel mutations, and substitution is also known as a point mutation. So you have one base pair that gets changed. So instead of an A and T, it is now a C and G on the top. The left is a wild type. The right side, you have a deletion of two base pairs, and et cetera, et cetera. So that's how you actually get it. The question is, how does this even happen? Yes, we get substitution, we get deletion, we get insertion and inversion. How is it actually happening? Who is introducing these mutations? When does it happen? How does it happen? We need to understand that stuff in order to find better treatments and to understand the mechanism of how these mutations are coming in the first place. So the left is no mutation. The types of substitution mutations or point mutations are silent, nonsense, and missense. If you're not familiar with these, please go to the textbook and read them. The understanding is that you've learned most of this in high school, and if you haven't, I know that all of you have gone to different high schools and had different teachers. I absolutely understand and respect that. So I'm giving you a slide to let you know what you should know from high school. If you don't know it, it's okay. You have your textbook, and more importantly, you have me. I'm here to teach you right from the ground up if you need it. You can walk into my office hours and say, can you go over silent, nonsense, and missense mutations? And I will go through it, all right? I will go through any high school material you want. It's all yours, and it's a personal lecture to you. So SNPs. What are SNPs? We just mentioned they're the most common type of genetic variation among people. They're single nucleotide changes. That's what they're called, single. It should actually be single nucleotide polymorphisms in pairs, because a double-stranded change is a mutation. But anyway, a single nucleotide polymorphism is a single nucleotide change. It can occur anywhere in the genome, but most of them are between genes. Between genes. Can you imagine if you had that many SNPs and they were all in your coding region? Like, that would be crazy, because that means you're going to have so many genes that are not functioning properly. So majority of them are between your genes, and they add to variation in a population. They don't add to disease. They don't contribute to disease. But many do. Many do, and those are the ones that when you get sequenced and go, oh, you have an increased chance of this, oh, because they're in areas that produce functional parts of the gene that can be associated with disease. And so most of these SNPs that you're getting throughout your lifetime, you're going to accumulate SNPs as well. You're going to be born with some, you're going to inherit it. Your mom and dad will give you SNPs, but you will also develop your own SNPs. So most have no effect on health and development. Some SNPs as you know, are associated with health and disease, and they can predict an individual's response to certain drugs. This is something I really find interesting, and maybe next term I'll present you with a paper that's so cool, and it's pharmacogenomics. That's the big area. Pharmacogenomics is huge. Understanding what drugs to give patients and how they will react based on their genome sequence. Instead of trying on a bunch of them and go, oh, it's not working. Oh, this one didn't work either. I know you went through 1,000 drugs and we tried everything. Let's see what works based on your genome. Susceptibility to environmental factors such as toxin. How susceptible are you? We can make a lot of predictions now based on your SNP patterns. Risk of developing particular disease, as you know. So let's look at the indel mutations. So those were substitutions. Let's move on to indel, insertion, deletion mutations. How did they happen? How many of you have learned about insertion and deletion? How does it happen? Oops, someone made a mistake. Who made the mistake? So look at the top strand. It's normal. The next strand, look at those being replicated. Look at that loop right there. What happened? Do you know what that's called? That's called slippage. It's called slippage. During replication, that strand, that top strand, it slips. And this happens in regions that are repetitive, either a bunch of T's, a bunch of A's, or repetitive sequences. So CAG, 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 they're more prone to slippage. And so look, when it slips, it creates a loop. And so everything just shifts to the left. And polymerase doesn't realize that. It continues to add bases as though it would. It can't see that loop. And repair enzymes don't see it either because that loop is very stable. It has to be with thermodynamics, which we're not going to get into. It has to be with the thermodynamic stability of that loop. It's stable. It just goes unrecognized. And then, when the whole strand is synthesized, if you stretch it out, you've got an inclusion of 3 bases that were not originally there. So this can have an effect depending on where it is, depending on where this mutation is. If it's in a coding region, if it's in a protein where now the amino acid sequence will change, then yes. It depends which amino acid is changing. So you cannot just look at that and go, I know the effect that's going to happen in the cell because of this. I know exactly what's going to happen. You don't know. Where is the mutation? What is it coding for? Et cetera, et cetera. So this is what we call backward slippage. And you can also have slippage in the template strand. The template strand can slip as well, and that's called forward slippage. So when we had forward slippage, I hope you can work this out. I'm actually telling you this. In other years, we just tell you to do it on your own. But you're stressed about the October term test. I don't want you to figure things out on your own. I'm just going to tell you a bunch of things. So that forward slippage leads to deletion. It leads to deletion, and I hope you can work that out. Just look at the way polymerase is polymerizing and why the backward leads to insertion and the forward leads to deletion. Another reason for mutations is due to what we call the formation of tautomers. How many of you have heard of tautomers? Tautomerization. One thing I want to mention right now is as you learn things in this course, in my lecture, in Beth's lecture, you will see that we're going to layer information on. We cannot teach you everything in one lecture. Oh, but when you talked about repair enzymes, you didn't mention this. Oh, but when you talked about DNA synthesis, you never mentioned this. I didn't mention slippage because we only have 50 minutes to go over everything. We're going to layer it. We're going to layer it one on top of the other, not because we didn't want to tell you, but because it was too much information. Just gather the information slowly and build it. Everything's connected. Now you know more about DNA replication than you did last class. Now you know more. Oh, slippage can happen. Not just mismatch. Not just mismatch. We can get slippage forward and reverse. That's something you can add to your list of knowledge. Things that you know about DNA synthesis. You see what I mean? We're going to layer it. And then second year will be more layering. Third year will be more layering. Fourth year you'll be layered up and ready to conquer the world. So thymine and adenine, right, these have a form, they have a structure, a chemical structure, and thymine, its normal form is keto. Adenine, its normal form is amino. So the keto and amino form go together. There is what's called spontaneous shift. It's nature. It's the way these bases react. That's how they are. They switch from keto form to enol form, for example. The thymine will switch from keto to enol, so you're like, so then what? Now that affects the Watson-Crick base pairing. Look at this equilibrium. The keto form is much more favored, not the enol form. So it's rare. It doesn't happen all the time, but it can. It can happen, and it does happen, and so you get the enol form. The problem with the enol form of thymine is that it prefers guanine, not because it's mismatched. Please understand this concept. This is not a mismatch. And again, a number one mistake, oh, there's a mismatch. It's technically, the enol form is not a mismatch. In your mind it is, but not in the cell. And that's why repair enzymes don't recognize it. They're not going to fix an enol and a keto. They're not going to fix it. They're not going to fix that enol thymine with the keto guanine. It won't be fixed. And so it goes undetected, and you can appreciate, now that we have this type of pairing, how this can lead to a point mutation. And so the same thing, another example of cytosine in the amino form is normal, pairs with guanine in the keto form, but in the amino form, not amino, it pairs with adenine. That's its preferred pairing partner. It prefers adenine over guanine. So understand that that's what happened. This is called tautomerization. The base pairs undergo another form called its tautomer form. Keto, enol, they're tautomers of each other. So look at this. Thymine, common form, pairs with guanine, the rare form. Common form of thymine can now pair with a tautomer of guanine, the enol form. And so this diagram simply depicts how you ultimately get a mutant when tautomerization occurs. Usually we get you to work it out yourself. It's an exercise and say, okay, this is what's happening. G star is the rare form of guanine, which is right there, the enol form on top, and that preferentially pairs with T. So instead of a C, you're going to put a T there, and now work it all the way through and see how it is that I get that mutation. Always keep in mind, that's one cell. The strands separate, DNA synthesis occurs again, becomes two daughter cells. Then again you get separation, you get synthesis, four daughter cells. If you just keep that in mind and track it, you will see how one of them ends up being a mutant cell. And I know that we brought this up a few times, I brought this up a few times. I never teach you stuff that's not that important. And you will appreciate this, maybe second term, when we talk about how cancer develops and how clonal expansion occurs. That's the clone. That's the clone that's a mutant. It's going to give rise to potential tumor. It all starts out with one cell, one cell. That can be a cell that's going to lead to variation in the population. If the enol form prefers thymine, then why does it not pair with thymine all the time? This tautomerization flips. That's what happens. It goes from enol to keto. So in the next replication round, it's gone back to normal. In the next replication round, it went back to its other form. On the exam, questions are not going to be, oh, but she didn't tell us that, this particular way of tautomerization leads to this particular type of mutation. You know tautomerization. So if the option says guanine has undergone tautomerization, go, yeah, that makes sense to me. Because based on that option right there, that's why that's happening up there. That makes sense to me. Yes, it must be tautomerization. There's no other explanation for this. Because you know that bases can tautomerize. You know that. All right, so how can the process of mismatch repair introduce a mutation? This is another layering piece. So we learned this in last class. So tell me how it's possible now for this process to lead to a mutation. It got fixed. It's a repair process. In other words, how can repair processes lead to a mutation? What do you think could happen? You don't know the answer because I haven't told you. I haven't taught you this yet. I haven't taught you this yet. You're like, well, how am I supposed to know that? You didn't tell me that this could also happen. But if you had to guess, how can the process of repair lead to mutation? Yes?
So maybe if there's like a frame shift mutation to the point that one of them is now mismatched with another because they got pushed over and it gets excised, that could be a really important or technical code on that as well.
Okay, so there could be a frame shift mutation, but we're not introducing other mutations in here. That's a good point. That could very well happen. But just as is this process, how could it introduce a mutation? Yes?
Like after the excision occurs and one of the bases might just polymerize.
Excellent. Very good. That is definitely one thing that can happen. Definitely. As the repair process is happening, when you cut at the bottom, because these processes of tautomization can happen at any time. It's spontaneous. That's why we use the word spontaneous. Boom, it happens. You don't know when. So during the repair process, you get tautomerization. And so polymerase adds it because the tautomer form favors is the key word, non-Watson-Crick. So they're not going to say, oh, it's going to want to take the mismatch because that's the favored one. The other thing I want to tell you, which you did not know, this is another layering, repair enzymes can also cut the template strand instead of the newly synthesized strand. That is a possibility. It can do that. Does it happen all the time? No, but it can happen. It can make a mistake, or the repair enzymes can have some sort of mutation that leads them to do this. So it can also be a mistake by them, not because of a mutation, just a mistake. Polymerase makes mistakes not because it's a mutant. It makes mistakes because it's polymerase. That's what it does. It has a certain amount of fidelity. Some mutagens are tautomerically unstable base analogs. Analogs are really things that mimic the original molecule. Thymine and uracil are common in structure. The difference is the methyl group and the hydrogen group. Here's what happens. The uracil can turn into 5-bromouracil. The 5-bromouracil is considered or looked as thymine. The cell looks at 5-bromouracil and doesn't realize that there's a bromine group there instead of a CH3 group. 5-bromouracil is a mutagen. It's a mutagen. So this can occur via tautomization. So you can go to 5-bromouracil via tautomization. And in general, just so you know, in cigarettes, they have this thing called 5-bromouracil. It's a mutagen. So wherever you are going to put a T, it puts the 5-BU instead. The 5-BU prefers guanine. The 5-BU prefers guanine. If the 5-BU preferred adenine, it wouldn't be a problem. It's shifted now for its preference, again, to guanine versus adenine, and so now follow this through and tell me why you get this mutation. Separates, and during that separation, it converts to its enol form. So it goes from keto to enol. The enol favors guanine. Now it pairs with guanine. So this cell is okay. This cell will not be okay because in the next replication round, this still has the mutagen. This one does not. So this one maybe could undergo apoptosis. Right? The cell could look at this and go, this is not right. Maybe it could undergo apoptosis. This one won't, because now it's properly paired as Watson-Crick, so it will not undergo. This is called a transition mutation. It's called a transition mutation because you went from one type of purine to another type of purine. If you go from a purine to a purine, that's transition. If you go from a purine to a pyrimidine, that's transversion. Here's a nice summary for you when it is considered transition mutation and when it is considered transversion mutation. So on a multiple choice, I could ask you, given this scenario, which type of mutation is happening? Is it transition or transversion? You would actually have to work it out. And just get into the habit of just writing it out, separating the strands, and then seeing how the next replication round will occur. Smokers' urine, if you look at smokers' urine, in the morning versus the evening, very, very high in these mutagens. And so the purpose of this slide is simply to say, if you are smoking, please do your best to quit. I know it's hard, but It's nothing good is coming from that. Here's data to prove it. Not that you needed data, I think we all know that. Transposable elements. This is what we're going to talk about next, our last topic. What are transposable elements? We have them. All organisms have them. These are regions of the DNA that move around your genome. They're called jumping genes. Have you guys heard of jumping genes? Jumping genes. And this was discovered by Barbara McClintock in the 1940s. And she discovered this in maize or corn. In 1940s, she said, guess what I've discovered? I've discovered jumping genes. They're genes that jump around the genome. And in the 1940s, no one believed her. Because when you come up with the concept called jumping genes, because everyone thought that the genome is just, it's stable, it's not mobile. The genes stay put, the sequences stay put in the chromosome exactly where they are. They don't move around. And then she called them jumping genes. And she was a woman in the 1940s doing science. So these were the two reasons why they thought she was crazy, and said, yeah, nice try. And then in 1983, she won the Nobel Prize. She was way ahead of her time, way ahead of her time. And what a phenomenal discovery. They discovered in the 1940s by her that jumping genes exist in corn, 1950s in Drosophila, 1960s in bacteria, and boom, 1970s in humans. Every organism has jumping genes. We call them transposable elements. What it is, it's a region of the DNA that moves. So it's called a transposable element. Transposable element contains inside it a region that codes for transposase, which is an enzyme that does the cutting and the pasting, or the copying and the pasting. It's cut and paste. It doesn't jump in the air, land somewhere. It doesn't do that. It copies and pastes or cuts and pastes in different regions. The way she discovered this, she asked a simple question. Why is this type of Indian corn, why are these kernels spotted? That's all she wanted to know. She was working in a lab and she was looking at corn and she's like, why are there spots on them? And that why question won her the Nobel Prize. The reason they're spotted is because there are genes that are jumping around. And they're jumping and interfering with the gene involved in pigmentation. So here is a gene for pigmentation. Here is a transposable element. It's a region of the DNA that got away from somewhere else, and it literally went right smack in the middle of that gene. Now, that gene cannot produce a pigment. So that's why you don't get color in that area. That's what transposable elements do, and she was able to discover this in corn. So how exactly does it work? Here you have the gene for pigmentation. If you have a TE in the middle of it, it can't produce the color, and that's why it's colorless or yellow. But if the gene is expressed, meaning that the TE is not in there, then it will be producing the enzyme that's involved in pigmentation. So that's where you get these patches. So in these patches, there are cells where the gene is not interfered by a transposable element. In the ones that are clear part, there's interference. And what's beautiful about plants is that their cells are they're stationary. And so you can actually analyze this really well and take a look at which cells have transposable elements and which ones don't in their actual interfering with that color gene. So there are two types, class 1 and class 2. Class 2 is the one that people talk about the most. And class 1 is the one that we have mostly in common in our cells in humans. So class 1 is RNA transposon, or retrotransposon it's called, and class 2 is DNA transposon. What is it made of? Transposon is this whole thing. It codes for transposase, which is an enzyme that it produces, and that enzyme will go then and bind to these terminal inverted repeats and then cut it out and then paste it somewhere else, or copy it and paste it somewhere else. The retrotransposon is actually going to produce an RNA molecule, and then it's going to reverse transcribe it into DNA and then incorporate somewhere else in the genome. So this can create a lot of copies. We humans have mainly retrotransposons. And ALU, if you've heard of ALU, that's the most common one. This is how it does it. That transposase gets produced, it binds to the ends, and then it cuts that part out, which is the whole transposon. and then transposition occurs. Transposition is when a transposon moves from one location to another location. So you get transposition occurring to the target site. The target site is in green, and then just goes and incorporates itself there. That's how the gene, quote unquote, jumps. During this process, you get what's called target site duplication. And that's how you add variation in an area of the genome, and you add duplication of that sequence. So it cuts this way, and then it splits it apart. When the double-stranded DNA splits apart, it looks like this. This gap has to be filled. It gets filled by polymerase. And look at that. Just follow this and ask yourself, oh, that duplicated now at the new site. There's 2 copies at the new site. It's called target site duplication. If that does not explain it to you well, then look at this one with specific sequences. So where the transposase cuts depends. If it's cutting 5 base pairs, or 2, or 3, or 10, that's going to be the size of the target site duplication. So transposase, what does it do? It cuts itself from the site and moves it to another site, and it also cuts the target site and opens it up for integration or for transposition. Transposase does a lot. Autonomous element is one that has everything it needs for transposition. A non-autonomous element does not have what it needs for transposition, but it can undergo transposition if it has an autonomous element present in the same genome. It can be anywhere in the genome, even on a different chromosome. So autonomous versus non-autonomous, they both move, except autonomous can do it by itself. That's what gets the word auto, by itself. And non-autonomous relies on the presence of another autonomous element in order to move. So retrotransposon, this is just an image from your textbook. They make a copy of the RNA, and then they reverse transcribe it, and then they insert itself. Imagine all the copies it can make, because all it involves is you're just transcribing that RNA molecule, and that's it. They transcribe the DNA, and then they make a bunch of copies. Endogenous retroviruses, we also have those. We have retroviruses. These are not viruses that are moving around and creating problems in your cell and infecting other hosts. No, it's not like HIV. These viruses once did infect our cells, but now they're dormant. They're just sitting there because they accumulated mutations. They can't move around. They can't do anything. They're just sitting in your genome, doing nothing. So this is how a TE can increase the copy number. Here you have your DNA. It's replicating. This is your famous replication fork. It replicates. It excises from here. It moves here. Then it uses a sister chromatid, the TE in the sister chromatid, to make a copy of itself. And now in this cell right here, when it divides, this one you're going to have 2 copies of the TE. And that's how copy number increases in a population. That's the mechanism. For retrotransposon, it's like this: transcription, double-strand DNA, integration. Imagine all the copies it can just keep pumping out. Transcribe, reverse transcribe, insert. Transcribe, reverse transcribe, insert. That's all it has to do. Transposable elements are most common in plants. If you look at the genome size, we talked about that C-value paradox and the C-value enigma a couple lectures ago. I asked you, why are the genome sizes so big in these plants? Look at that. Barley, 85% of its genome is transposable elements, jumping genes. 85% of it. It's accumulated so many over time. So if you compare barley, corn, and rice, if you have 3 genes, look at rice. The genes are much more compact. They're more together. Look at barley. It's got all these black elements. Those are all transposable elements, and they're all in between genes. They're not interfering with the gene, but they've accumulated over time because of the amount of replication that's occurring, amount of copy and paste. That's why the sizes of the genomes of these plants are so big. Humans, 50% of the human genome contains TEs. So you're like, I don't understand. If TEs are going and they're jumping around, then we should be dead by now because they're going to interfere with all of our genes and the function of the genes. They don't, but we have them. And sometimes they do. And that's how you get diseases. That's how you get some types of cancer, is through the insertion of a transposable element, such as ALU, into the gene that's very important for function. So 78 to 80% of the human genes contain TE, up to 1,000 per gene. Here's a gene with exons, and look at that. Look at that. All these colors are transposable elements that have selectively inserted themselves in areas that are called safe havens. They don't interfere with the function. They don't interfere with the making a functional protein. Most TEs in our bodies are dead. They're dead due to inactivating mutations. They're dead. They can't undergo transposition. And those that can do, we've developed a process to silence TEs. And sometimes that process of silencing TEs goes aberrant, and then we can get cancer. So sometimes that does happen, and you will read journal articles about alu elements. These humans have over 1 million copies of alu, all in our genome. So in case you're in bioinformatics, and you ask the question to your summer supervisor that you're going to go work with and volunteer with, you can present this stuff and tell them, I want to work in bioinformatics, and you understand transposable elements, you understand why the genome is so large, what do we make of it, is it junk, what does it mean? This is what it all means, okay? So on that note, I will see you next class.